Hi everyone. I'm Dan Maloney, and I'm an application scientist here at BSI. And today we're going to be talking about our new tool. It's called Peaks Glycan Finder, and it's a software platform for deep glycoproteomics with structure resolution. And just a quick outline of what we're going to be talking about today. So first, I'll just give a quick overview of what glycoproteomics is and some of the unique problems that are specific to analyzing mass spectrometry data from glycoproteomics. And then I'll be talking about how Glycan Finder can help with the problems such as site ambiguity, structure ambiguity, and how we can also have built in some label-free quantification into our glycoproteomics tool. Yeah, and just like everything with Peaks, we provide the results in a very intuitive way for you to understand. So we'll be going through the actual result reporting in terms of glycoproteins and glycan profiling. All right, so Glycan Finder was designed with a few questions in mind. So first, we want to answer the question, what glycoproteins are actually present? And then once we're able to do that, then we can then figure out what are the composition of those glycans and the location of those glycans on the protein. And if we can then do that, then the next question that we want to answer is there a difference in the abundance of those glycans between conditions? So we've designed our interface to provide the answers to these questions in the most efficient way possible. Yeah, so just a quick overview of glycoproteomics. So cells synthesize a wide assortment of glycoproteins with a wide variety of structures. And that's the most important point when it comes to analyzing mass spectrometry data from glycoproteins, because of those wide variety of structures, and because the glycan can actually fragment itself when creating the MS2 spectrum, it can make a very complex spectra that's difficult for the software to actually get a correct sequence from. But it's very important that we work with glycoproteins because glycosylation has been shown to be involved in a wide variety of biological processes, including cellular development, cell-to-cell communication, and immunity. So Glycan Finder at this point deals with two types of glycopeptides. So there's N-linked glycopeptides, which attach to the asparagine in a very specific binding motif. So the actual location of an N-linked glycopeptide is not that challenging to identify, but the structure of the N-linked glycan can be complex, and there's a wide variety of different N-linked structures. So dealing with actually finding the correct composition and structure is a challenging problem. O-linked glycans are different in that the structures can be a little bit more simple than N-linked glycans, but they attach at a wider variety of positions. So they attach at the serines and threonines, and there can, in some cases, be several different sites on one particular digested peptide that an O-linked glycosylation can attach to. So actually finding the location of it can be a challenge, and Peaks has some ways of dealing with that. So in the past, there's been several different glycoproteomics tools that have come out to tackle this problem, and they can generally be put into two categories. There's peptide-based glycoproteomics tools, and there's glycan-based proteomics tools. Glycan Finder is actually a combination of both, which allows you to get the best of both worlds. And I'll show a little bit about what I mean about that. So here's just a very high level breakdown of the Glycan Finder algorithm. So we deal with LCM-SMS data, and first we feed the data into a spectrum classifier. Of course, we take the MS1 data and do peptide feature detection, where we're looking to find the area under the curve of the peptide feature from just the LCMS details, and then use that data later on for quantitative purposes. And with the remainder of the spectra, we then classify them into glycopeptide spectra, potential glycopeptide spectra, and non-glycosylated peptide spectra, because glycopeptides have a very specific signature in the high mass range that looks like a glycan, and we just take all the spectra that look like they're glycans and put those into our glycan finder algorithm. With the remainder of the spectra that look like unglycosylated spectra, we feed those into a typical PeaksDB search, identify unglycosylated peptides, and from that we get a candidate list, a list of candidate proteins that we can then feed into the glycan finder search to assist in the list of actual proteins that are present there. As well, we can then research those peptides if they have a motif that looks like a glycan, or potentially a signal that looks like a glycan, but might not be as strong as what was originally found in the spectrum classifier, and maybe save some glycopeptide identifications that way. So once we have our search space for our glycan finder, we can then identify glycopeptides and glycoproteins and do FDR estimation on those as well. We also have the ability to do glycan de novo sequencing. In that case, what we're doing is we're trying to find a list of potential spectra that are possible identifications in. For those of you who are familiar with Peak Studio, we have the de novo only view, where you have a list of good quality spectra that were partially matched to a protein sequence by de novo sequencing. And that can provide you a list of something where you can potentially identify some novel sequences. So we do that for you as well in Glycan Finder. All right, so with this approach, we're able to provide the best of both worlds in terms of peptide-centric and glycan-centric searches. Say, for example, with a peptide-based approach, if you have a good glycan and you have a good peptide in the actual spectrum, you're likely to get a very good result. But with a peptide-based approach, if you have a poor glycan signal, then it's less likely that you're going to be able to get a good result. But if you have a poor peptide, you might still be able to get a good result that way. The opposite's true with a glycan-based approach. So if you have a poor peptide signal in that case, you're probably not going to get a good identification. With Glycan Finder, since we use a combination of both approaches, we're able to maximize the sensitivity of the search and find potential matches if you have a poor glycan or a poor peptide, in order to get the most glycan matches possible. Right. So one of the first problems that we have to deal with in terms of identifying glycopeptides is site ambiguity. And what do I mean by that? It means, what do you do in the case where you have multiple sites where a glycan can attach, such as an Olink glycopeptide, where you attaches to threonines and serines. And take, for example, this peptide here, where you have 1, 2, 3, 4, 5, 6, 7, 8 possible sites where a glycosylation can happen in this case. So localizing a glycosylation on this spectrum can be quite difficult. So one way to actually measure whether or not the localization is correct is by using an A-score. And this is what we've typically done in Peak Studio, where we look, we provide the A-score as a -10 log P, where generally speaking, a p-value of 0.01 is equal to a -10 log P of 20. So generally speaking, 20 or greater is a p-value of 0.01 or less. Or in the case of, like say, a 0.05 p-value, that would be equal to a -10 log P of 13. So in this case, we have reasonable, so with a p-value of 0.05, we're able to localize the glycosylations to these two sites, which is quite reasonable considering the high amount of sites where the actual glycosylation can happen in this case. So what the A-score is doing is it's considering all possible matches, considering these glycosylations and this peptide. And if you compare the sequence alignments of those different possibilities, how likely is this assignment where you have the glycosylation on the first and the second threonine here? How likely is that compared to the other possibilities? So it is quite likely in this case because we have good fragmentation showing on T1. We have a glycosylation which is supported by the fragment ions here. Again, this is ETHCD data, and we also have good fragment ion support for a glycosylation on position 2. So considering those, we're able to fairly confidently localize the glycosylations to these two sites. And by providing the A-score, we give a way to actually localize glycosylations in a high throughput way to make the most glycopeptide localizations that you can. All right, and now for the concept of structure ambiguity. So in Glycan Finder, we provide a way for you to determine what is the most likely glycan structure that matches up to a spectrum. And this can be a very challenging problem because you have multiple glycans with the actual same mass and the same composition, but with a slightly different structure. And Peaks provides an S-score. So it's a normalized score gap between the most, what we consider to be the actual structure that is matched to the spectrum. Using our Glycan database, which is a database based on a manually curated structure database. And we determine that this is the most likely structure that matches up to this sequence. And what we determine is that it's 16% more likely to the next best match, which is this structure here. And the reason why we consider this to be the more likely structure is because you have these fragment ions that show that this is the only structure that's possible if you're comparing these two structures here. These fragment ions would not exist if this was the actual structure that was found in this spectrum. So by doing this, we were able to provide the most likely structure for your sequence. All right, and about that structure database, so we provide some built-in Glycan structure databases, but you can also provide your own manually created structure database using some public database structures. So here's an example of one of the Glycan databases that we provide in Peaks, where we have HNAGF, it's for the hex, hexNAC, and so on and so forth. And it's provided in a series of just letters and brackets to represent the sequence. And here's an example just to show how this works. So starting from the left to the right, you're going from where it attaches to the amino acid forward. So you have the hexNAC that actually attaches to the peptide, and then you have in brackets an F indicating that we have a fucose that's coming off that position here. And then the next bracket would then show where you have the next hexNAC and how that attaches to hexoses. Shows how you set up the actual the brackets shows what branches off. So you have like an H within one bracket here. It shows that that's one branch off the first H. And then the second H in another set of brackets shows that this is also attached to here, which shows that we have two H's attached to the N here. And that's just a quick breakdown of the structure. And by doing that, you're able to create your own glycan structure database. All right, and we also provide some FDR estimation. So for the actual unglycosylated peptides, we have our typical target decoy database approach, where we take our target protein database and shuffle it, and then append those random peptide sequences to our target database. For glycans, what we do is we create decoy peptides as well as decoy glycans, actually making decoy spectra. What that means is we create decoy glycopeptide spectra where we have randomized peaks in the area where you'd expect to see the actual glycopeptide. And by doing that, you have a decoy glycopeptide spectrum. And when we do this, we are more stringent with the requirements for our decoy glycopeptides than we are for our target peptides. What I mean by that is, in order to actually identify a target peptide, you need to have it hit in the target peptide sequence and the target glycan database. To match a decoy peptide, we only need to identify a decoy peptide or a decoy glycan. So the requirements at that side are more stringent than the requirements for the target database are more stringent than the decoy database. So likely we're overestimating the FDR, but in that case, we're providing more accurate results. All right, so let's actually get into the result reporting. So the result reporting all goes back to those first questions. First question being, what is the list of glycoproteins that are actually present in the sample? And that's the first thing that we report, and we report it in an intuitive way, where we take the coverage view, typical to Peak Studio, where we have blue bars showing matches to our sequence. In this case, we're showing glycopeptides in like a purpley pink color here, showing where the actual glycopeptides are attaching to the protein. And then in the associated peptide list, you have a list of the glycan structure compositions, sorry, the glycan compositions that are attached to the peptide, as well as some quantitative information. In this case, it's just 3 replicates showing the actual sum of the area under the curves of the peptides that are associated with these positions. Next, we want to deal with the next question of the location, composition, and amount of glycans we find on the protein. So here we provide a glycan profiling view, where if you take a look at all the glycans that are associated with a particular position, take a look at all the glycans that are associated with a particular position and provide in a pie chart out of the total area of all the peptides that occur at that position, how much of that total area is associated with each composition, to give you an idea of which glycan is most abundant at that position. And then the glycan profiling view, we give you a glycan XIC across the different samples. In that case, to give you an idea of how reproducible that area is. In the case here, where we just have 3 replicates, you can see that this glycan is quite reproducible across the 3 samples. All right, so now let's get into some application examples. So what we're going to talk about here is the case of just 3 samples where we're looking at IgG subclasses. So there's IgG 1, 2, 3, and 4, and the main sequence difference here actually occurs where the glycan attaches to the protein. So in order to actually accurately separate these 4 different subclasses, which are difficult to actually separate by LC, we need to actually be able to get some good glycopeptide identification. So Glycan Finder is able to do this pretty accurately. So you can see here that we have some good spectra showing the peptide that would allow us to separate IgG 1, 2, 3, and 4 here. And we provide that for you in an intuitive way. And also because we're doing some label-free quantification, we can give you an idea of the approximate amount of each subclass in the data set. So it's almost like 55% for this, 10% for IgG3, 30% for IgG3, 10% for IgG4, and then 5% for IgG2. So it gives you an idea of the amount of the different subclass types in our samples. And then dealing with the amount of the different compositions, we are able to look at these pie charts from the different samples, or actually from the different subclass types, and see the amount, get an idea of the amount of the different glycan compositions that are occurring in our samples. And then as well, we provided in another view here, which I find that this is a very intuitive way to look at the data, where we provide you a list of all the different glycopeptides, a suggested structure for each of these, and we also give in a bar chart the area under the curve of the peptides that are associated with each of these different compositions here. So since these are replicates, you can see that the area under the curve of the peptides that are associated with these glycans are pretty reproducible across the different samples here. Yeah, and with that, that's all I have for the actual slide section. Before I get into the actual software demo, I'd just like to thank the development team that's worked on Glycan Finder, and also our collaborators at Radboud and University of Waterloo. All right, now let's actually take a look at the software. All right, so this is Glycan Finder. And this is also built on the new Peak Studio platform, which we'll be releasing shortly. So you can see for those of you who are familiar with Peak Studio, it looks very similar, but with a splash more color. So everything's still in the same places. So up in the top left, you can create new projects. Go here to the configurations, and you can set up your databases. And very importantly, in this case, glycan databases. So let's click here, and you can see that we have some built-in glycan databases. So we have N-linked, O-linked, specific to yeast, mouse, and human IgG here. So those are the built-in databases, and these are provided in GlycoCT format, but we also accept GDB glycan databases. And then in terms of your actual target protein sequence database, those are added in the same way from this list here. And then if you want to create a new project, you go top left, click this button here, click here to add your data. And I'm going to add the example that we've been talking about so far. So we have just 3 replicates of human IgG, and this is from a Q Exactive. So you load those in, and remember that we have some label-free quantification built into Glycan Finder. So if you do have replicates or different conditions, you want to load those as different samples. So take 1, 2, 3, add it over to the right, and then we're telling Peaks that these are actually different replicates or different conditions when we go to do some quantification later on. Specify the instrument type, and fragmentation, digest, and then you can copy that to the whole project, then continue on to the workflow section. So we have 3 workflows that are built into Glycan Finder. So N-linked and O-linked is when you're specifically looking for those 2 different glycosylation types. If you are looking for O-linked glycosylations in a sample where you expect to find both, we do recommend doing the N-linked followed by O-linked search. So what this does is it first searches for N-linked peptides, which require less of a search space because of the reasons we talked about in the slides. So basically what you're doing is you're first storing all the spectra that we know are matched to N-linked peptides so that we limit the search space for our O-linked search, which requires a much larger search space. In this case, we're just doing an N-linked search, and then you do data refinement, and followed by our Glycan search. So here, I'd just like to talk a little bit about some of the options here. So you have the option up here to search if it is enriched for glycopeptides. You click that button there, and it will use a larger search space in order to maximize your identifications from an enriched data set. You can also click this button here, which will only identify glycopeptides. And then you just have your error tolerances. The only difference here is that we have a error tolerance that's specific for the glycan fragment ions. And then specify your PTMs. So I'd just like to point out what's most important here is that these are the PTMs other than glycosylation that we're looking at. So just set your typical PTMs that you'd expect for any typical proteomics search. You'll notice that we do have a list for glycosylations, but of course, since it's Glycan Finder, it is searching for all the potential glycans that are found in your actual glycan structure database. Yeah, and then specify the adducts that you would allow, as well as click this button to specify if you're going to include a search for fucose and the maximum number of fucoses that you allow per peptide. And then this area here is important because this is where you specify the search settings for your quantification. So you click this button here and you set up your grouping for quantification. So by default, it'll just set every sample up into its own group. If you remove all, and let's say samples 1, 2 are from the same condition, you can put those in a group like so, just like you're doing a label-free quantification. In this case, I'm just going to set them all into separate groups because we only have one group in this case. Yeah, and then set your target protein database, and that's based on the workflow that you select. And click report, and it'll get you to this page where you can set your report filters. So one of the new things about the upcoming Peak Studio, and as well with Glycan Finder, is that you're able to set your result filters before the search. So in this case, we want to select a 1% FDR, but you can also provide set your results based on what we call a glycopeptide score, a peptide score and a glycan score. So basically, this is just an ion score, meaning that it's just the log transformed the log transformed sum of the intensity of the fragment ions that are actually matched to our glycopeptide. So it's basically just the amount of actual fragment ions that match to our expected sequence. In this case, we want to use the FDR, then click finish. It'll run your results. So once you get your results, this is what they'll look like here. So get some result statistics showing the number of glycopeptides, other peptides are matched to our sample. the mass error distribution of the unglycosylated peptides and the glycosylated peptides, and a list of the different glycan compositions and predicted structures that we've matched up in our sample. Then you come here to the protein tab, and you can see a list of the different glycans that are the different glycoproteins that we've identified here. So you can see that we were able to successfully separate our IgG1, 2, 3, and 4 here. And then if you look at the actual coverage view, you get our nice coverage view, our nice pile up, where you can see all the different glycan forms that are matched to this protein sequence. And over on the right, you can see a list of all of the different compositions that we've matched here, sort by number of spectra, and you can see which glycan compositions had the most MSMS spectra that were matched to it. And some nice tools that are slightly hidden, but if you click this button here, if you actually click on the amino acid where the glycan attaches, you get that pie chart where you see out of the total area of all the peptides that are matched up to this, which glycan compositions are most abundant out of the total. Then you click this button up here in the top right, glycan profile, and that gives you this view here where you can see for each of the different glycans, you can get the area under the curves of each, and a reported XIC where these glycans are actually found in terms of the retention time. And you can click this button up here at the top, click this button to deselect all. Then you can find, say, one glycan that you're particularly interested in. There it is. You click there, and that will show you all the details that are specific to the glycan that we're interested in here. So here we're looking at a HexNEC5, Hex5, and a 1 fucose, and then you can see the percentage out of the total area that's assigned at this position, how much of that total area is actually assigned to our particular glycan, and get a look at the XIC and see if that looks nice too. And then, yeah, of course, if you're just want to work just strictly from text format, if you want to put this all into a CSV file for further processing, you can click this button up here in the top right and say save to text format. Click this button to say export glycan profile for all proteins, and it'll create a table with the areas, the compositions, and all of the appropriate information to do some post-processing in a CSV format. And another nice thing with the latest version of Peaks and Glycan Finder here, click this export button, and you can see all the different CSVs that you can create from this. And a great thing about this is that you don't even have to open the result view to get these exports. You can just double click the export button and quickly generate some CSVs for further processing. Some additional features that we have here. So we have this partial tab, which I'd like to talk about in a little bit more detail. So this partial tab is interesting because what it gives you is it gives you spectra that have a good signal from what looks to be a glycan, but in some case, you don't actually have a good signal from the backbone peptide. And what we then do is we say we have a peptide with a certain mass, but we can't confirm for sure what it actually is because it just didn't have a match that's within our 1% FTR cutoff or several other reasons. But we have a spectra with a good quality signal coming from a glycan, but no peptide sequence assigned to it. So you can use this list to do some troubleshooting or just to look for novel glycans here and see what you could possibly find from this, because you can see here, we don't really have a good signal in the range where we'd actually match to a peptide. But we have a good glycan signal. So what is this? It just requires a bit of manual work to figure out what that is. And that's what this list provides you. And you also have the feature tab here where it shows you more focused on the LCMS data. That's what we mean by feature. So it's the LCMS peptide feature is what we're looking at here. So where all the data is stored in a feature list where we have everything associated with this LCMS feature, the area under the curves of the peptide here, the glycan composition, and you can work from this table instead as well to look at all the potential features that are assigned to our data. Yeah, and that's Glycan Finder. So we have a trial of this available now, so we can send this to you. And so this is available for several different data types, mainly for timsTOF, Orbitraps, and also XenotOFs. And yeah, if you'd like a demo, let us know. And if you have any questions, please go ahead in the chat.